Let's go through a monohybrid cross like that we saw in Mendel's pea experiments. So let's use the flower color example from Mendel's peas experiments. So capital P is going to be a purple flowering pea plant, and then lowercase p is going to be a white, right? So that's our dominant color is purple, then our recessive color is white. Let's first look at this term monohybrid. So the word mono means one. So in this case, we're only going to be looking at one character. That character for this case is what color are the flowers? And then the term hybrid means heterozygous. So all of the parents are going to be heterozygous for the one character we're looking at. So that means that I know the genotype of the parents. They're going to be big P, little p. That is a hybrid. So let's go ahead and look at this cross. So our parents are going to be big P, little p, crossed with big P, little p. So we can tell that both of these parents are purple flowering plants, right? As long as you have one capital P, it covers up the lower recessive P, and that's going to give them a phenotype of purple. The first thing we have to do before we can figure out the size of the Punnett square is look at the gametes that these parents are going to produce. So this parent on the left can produce either a P, a big P, or a little p. So they have two different possible gametes. Same thing for this parent on the right. So this parent can produce either a big P or a little p. Those are all of the possible gametes we can make. Since each parent can make two different gametes, remember the gametes are eggs or sperm, we're going to need a 2 by 2 Punnett square. So one parent goes on the left, typically mom, but it doesn't really matter. So I'll put one gamete in this row and one gamete in this row. So big P, little p. The other parent goes on the top. So this time I'll put one gamete in this column and then one gamete in this column. Big P, little p. And then you just drag the values from the outside to the inside. So this P and this P. So we end up with big P in the first box. In the next box, we're going to pull big P, and then little p comes from the top. Down here, we've got a big P from the top and a little p from the left. I am going to write the capital P's first just to keep everything the same. And then this box gets a little p, little p. So we're going to look at the genotypes and the phenotypes of the offspring. There were 3 different possible genotypes. Genotypes are the actual alleles. So we had one that had big P, big P. There's 2, 1, 2, that end up with big P, little p. And then there's one offspring with a genotype of little p, little p. So this is the genetics, with the genetics that the individuals actually carry. What we can actually see is called the phenotype. We're going to end up with 3 purples. And those are coming from these 2 individuals. And then we end up with 1 white. Remember, the phenotype is always the physical trait, whether it's purple or white. So this gives us a 3 to 1 phenotypic ratio. This is the standard expected phenotypic ratio for all monohybrid crosses. So anytime you see a monohybrid cross, you should expect for the outcome to be 3 of the dominant trait, which in this case is purple, and 1 of the recessive trait, which in this case is white.